Welcome back to my presentation. We will now turn our attention to a third important data type in ecology. These are quantitatively marked patterns. In this case we have a univariate pattern, but our data contain additional information on each point. This information is in general called mark. Here we have a specific type of mark, a continuous or quantitative mark. The analysis of quantitatively marked point patterns assumes that a pattern was created in two steps. A first process creates the univariate pattern, for example, animal droppings. A second process creates the continuous mark, for example, the number of seeds of a given species. Note that we can have also two marks. For example, the first mark will be the number of seeds of a given species 1, and the second mark the number of seeds of a given species 2. And this is the case for José's data of animal droppings. A quantitatively marked pattern could represent many different things. For example, the mark could be the size of the trees, number of flowers, number of nestlings, or number of seeds in droppings. Again, in this type of analysis, one is not interested in the spatial structure of the univariate pattern, but on the spatial structure of the marks, conditionally on the univariate pattern. Therefore, typical questions asked in this type of analysis are the tree sizes randomly distributed over the trees? Do small trees group together at small distances? Are small trees typically close to large trees? Or are seeds of species 1 typically placed close to seeds of species 2? Therefore, again, we can ask for these typical spatial scales where the number of seeds show positive or negative correlations. In the last overhead, I explained the data structure and typical questions for quantitatively marked point patterns. Now our task is to develop suitable summary functions and null models. Summary functions that are adapted to the conditional nature of this data type are called mark correlation functions. What is now the idea of mark correlation functions? They are very similar to the idea of mark connection functions I presented in the last video. Again, we look at point pairs that are separated by a given distance. And again, we estimate the value of a given test function of the two marks. For example, this test function could be the product of the two marks. In our case, the first mark is the number of seeds of a given species 1 in the first dropping, and the second mark is the number of seeds of the second species in the second dropping, which is located a given distance r away from the first one. The mark correlation function is then the mean product of m1 times m2 taken over all pairs of tropics that have this given distance r. Of course, there are many more mark correlation functions possible, because one can think of many different test functions constructed from the values of the two marks. Under random labeling, we have spatial independence of the values of the marks. For example, the so-called R-mark correlation function is the mean value of the first mark of the point pair. Note that this value may be in general different from the mean of the first mark because it is a conditional mean, with the condition being that a second point is close by. So for example, in case of competition, it could be that trees are smaller than expected if they have a second tree nearby. The mark variogram, for example, is the square difference between the two marks and closely related to the geostatistical variogram. Each mark correlation function captures a different feature of the data. Which function or which mark correlation function to be used depends on the specific research question asked. The suitable null model is again random shuffling of the marks, but now this is done in a way that it depends on the very specific data structure. So please look at my book for more detailed data types and null models. In case of José's data, the seeds are transported in the droppings. So we would need a null model that shuffles entire droppings among the observed locations. Thus, the observed co-occurrence of the seeds inside the droppings must be conserved in our analysis. Now we have all the theoretical knowledge to analyze the data of José. We have the univariate pattern of the droppings of the animals. We have two different marks for each dropping, the number of seeds of species 1, the number of seeds of species 2. And in the bivariate case, we look at the mark of species 1 in the first dropping and the mark of species 2 in the second dropping. In the next step, I will present you how to analyze this data set by using my software Programmita.